Hello friends, welcome to my channel and I hope you like my videos. So today in this video we will discuss on centrifuge basics and you can learn what is centrifuge and its types and how to operate it with other details, right? Friends, my YouTube channel name is Pharma Quality, so please view, like, comment and subscribe my channel. So let's start with the topic. So now here we will discuss about the definition of a centrifuge. So a centrifuge is a laboratory or industrial device used to separate components of a mixture based on their density by applying centrifugal force. And it operates by spinning the sample at high speeds, causing denser particles to move outward and lighter components to remain closer to the center. So basically, this centrifuge follow the sedimentation principle and Stokes law where relative centrifugal force depends on speed, that is in rpm, and rotor radius, right? So it is widely used in the biotechnology, pharmaceutical and diagnostics for tasks like blood separation, DNA extraction and protein purification. And into the pharmaceutical industry there are several test solution you need to centrifuge it, and you need to take the supernatant solution for further analysis and testing purpose. So overall, the centrifuge is work based on the principle of sedimentation, right? So now here we will discuss the different types of centrifuges which are used into the different industries. So first one is the bench top centrifuges. So bench top centrifuges are compact device commonly used in clinical and research laboratories. Actually, they provide efficient separation of biological samples such as blood and cell cultures while saving the space because it is bench top. So you can easily accommodate on the bench top, right? This centrifuge operates at variable speeds and accommodate different rotor types for diverse applications, right? Next one is the high speed centrifuge. So it can reach higher rotational speeds, which making them ideal for molecular biology and biochemical application. They enable the efficient separation of cellular components like nucleic acids and proteins. So it generate greater centrifugal force for precise sample processing, right? Now next one is the ultracentrifuges. So ultracentrifuges operate at extremely high speed, which enabling the separation of very fine particles like viruses and cellular organelles. So they generate immense centrifugal force, allowing precise fractionation of biomolecules for advanced research and analytical applications, right? Next one is the microcentrifuge. So microcentrifuges are designed for small sample volumes and are commonly used in DNA, RNA and protein studies. They provide rapid and efficient centrifugation for molecular biology and biochemical applications, right? Next one is the refrigerated centrifuge, right? So refrigerated centrifuges are equipped with temperature control to maintain sample integrity during separation. They are essential for processing temperature sensitive samples in biological and biochemical research and also in the pharmaceutical industries, right? Next one is the industrial centrifuges. So these are used in large scale application which include the pharmaceutical and food processing industries because they enable high capacity separation of liquids and solids, ensuring the efficiency in manufacturing and production processes. So these are overall the different types of centrifuges, right? So now here we will discuss about the overview of components for centrifuge, right? So we'll discuss the major parts into the centrifuge machine. So you can see here is a latch, it is called, which is a locking system, and this one is the lid. So these both are used to secure the chamber to ensure the safe operations. So once you will need to run the centrifuge, you need to close this lid with the locking of this latch, right? And this one is the chamber which encloses the rotor and samples during centrifugation, right? And here it is the control panel which allows the user to set the speed, time and temperature, right? and here is the rotor. So rotor holds the sample tubes and spins at the high speed based on the rpm set into the control panel, right? And here it is the motor. So motor will generate the powers so that rotor rotates for effective separation, right? And here you can keep the sample, so the substance that is being processed for separation. So these are overall the major parts into the centrifuge machine, right? So now here we can see that how centrifuge machine separate the denser and smaller particles by centrifugation, right? So for example, if you're putting the a mixture of sample here into the centrifuge, so based on that speed and temperature, it will generate the centrifugal force, and the denser particles will remain here and the lighter particles will remain here. So this one is the called pellet, and this one is the called the supernatant. So based on this it works, right? So now here we will discuss what are those factors which can affect the centrifugation efficiency, right? So for the first one is the rotor type. So fixed ankle rotors allow faster sedimentation, while swinging bucket rotors provide better separation for gradient centrifugation, right? So rotor plays the very important role while affecting into the centrifugation, right? Next one is the speed and relative centrifugal force. So higher the centrifugal force improves the separation, but excessive force can damage the delicate samples, right? Next one is the sample volume and density. So proper balancing and selection of tubes prevent the operational issues, right? So once you are putting, for example, you are having the one sample, so into the next, opposite of that well, you need to put the same mass of sample into that to balance the sample, right? Next one is the temperature sensitivity. So some biological samples require refrigerated centrifuges to maintain the stability. So based on the requirement, temperature need to be set into the centrifuge machine, right? So now here we will discuss the operation procedure, that is how to operate the centrifuge machine, right? So first one is the preparation. So place the centrifuge on a stable vibration free surface and ensure it is properly connected to a power source, right? So check that the rotor is also clean and free from dust to prevent the contamination or imbalance, right? Next one is the sample loading. So load the tubes symmetrically into the rotor to maintain the balance and avoid excessive vibrations, which ensure all tubes contain equal volumes to the prevent uneven distribution of force during spinning, right? Next one is the setting parameters. So adjust the required speed in RPM, duration and temperature based on the type of sample and separation requirements. For this, you can refer to standard protocol for optimal settings, right? Next one is the start option. So secure that the lid properly to prevent accidents. That is, lid should be closed properly. Then press the start button. Some centrifuges may require an additional safety checks before initiating the spin cycle, right? Next one is the monitoring. So monitor the centrifuge closely for abnormal vibrations or noises or any error messages. If any issues arises, stop the machine immediately and inspect the rotor and sample placement, right? Next one is the completion. So allow the rotor to come to a full stop before opening the lid to prevent any spillage or sample disruption due to residual motion, right? So it should be stopped properly. Next one is sample retrieval. So gently remove the tubes, ensuring that minimal disturbance to the separated layers. Handle the samples carefully and store them as per the experimental requirements to maintain the integrity, right? So these are the overall operating procedure, that is how to operate the centrifuge machine. So friends, this is overall the basics of centrifuge machine. Thank you.